110 decibel, you will become completely deaf after 1 hour in that thing. So you need protection. And then it's of course, in case of an experiment, the person needs to look at the display. There is also a prism glass there, because you need to, I mean, the person is looking upright, so you need to do something about it so that the person can see the display. And of course, you cannot use, in the past, people were using CRTs. You can't use a CRT. The image will completely be corrupted because of the magnetic field that is exiting the coil. Also, metallic components can be in the vicinity that will be drawn into the coil. So it's not an easy setup. And usually you do it with a video projector so that you can keep distance, and the image is being projected onto a screen. So it is a lot of precaution there. And yeah, if you go to look at slide 36, you see these anatomical static images that can be taken from the brain at different angles or different cuts through the brain. And then you, in slide 37, you show this experiment here that people usually consider when to figure out if everything is working properly. They have this checkerboard that is flickering, and if you look at the checkerboard, you will really become a bit dizzy. So that checkerboard is being switched on and off, shown here, that regime, 60 seconds on, 60 off. So it's a flickering thing. And then if you click further on slide 37, you see then that blurry image that is being picked up. And then eventually you actually have a region of interest indicated by the red square. And the activity in there is being monitored. If the checkerboard is off, then you have low level of activity in that area. When the checkerboard is switched on, you have a higher level. So the level of activity is periodically changing in sync with the checkerboard being switched on and off. So this is then sort of evidence that you can use to build your brain-computer interface on. So if you look at slide 38, these are the sort of recordings then. You have a paradigm, and indicated by the gray, blue, dark gray bars here, that stimuli that being shown in a certain sequence. And you see the bold signal evolving according to, in response to them. Sometimes it goes up, sometimes it goes down. And the lower signal, the blue one, the blue signal is actually the hemodynamic response function. This is the function that's being used in order to interpret or statistically process the recorded data. Of course, the scale is arbitrary. If you express it in percentage of change with respect to baseline, then you're looking at a signal amplitude of about 0.5 to a few percent. So the signal changes are also pretty small, a bit like in the case of EEG, very small changes. So you need to be aware of that. Probably you need to repeat your paradigm also a few times to figure out what the response is. So if you then set up that you want to use in terms of communication, slide 39, you can have like two conditions. Condition one, you record some activity in response to that. Then you can present condition two, you can record some activity response to that. And eventually, for your region of interest, you can figure out the signal change for the red case and the green case is differing, and this way for your region of interest, that is, and this way you can figure out the response pattern, the responses to your paradigm, for example. It is not an easy task, as you can guess, and it's extremely slow. You take a brain scan every 2 seconds or so. So if you have to collect a number of those, so you can communicate maybe 1 to 2 characters per minute with this kind of technology on that order. So it's extremely slow, but it is something that is used for coma patients that cannot actually anymore communicate properly or not at all, then there is a way then to figure out if they're conscient or not. Sometimes it's difficult to figure out if your coma patient is still conscient of the environment. So this way you can ask your patient to think about tennis or about chess or about whatever, and see if there is any shift in activity in the brain in response to that, and if that activity corresponds to particular areas of which you know that are related to motion activity or to arithmetic activity. This way you can figure out if your patient is conscient or not. Slide 40, you see here a NIRS, near-infrared spectroscopy. It's a bit difficult to pronounce. Here is still another principle. You actually are also looking at a sensor that is, but this time it is a light sensor, transmitting light through your skin, through the skull, into the brain, where it gets absorbed, and part of it reflected. And the reflected signal is picked up and related to what was originally sent. So not so easy to do. Indeed, to slide 41, you see the principle. So light is shining of different wavelengths, wavelength range between 700 and 1000 nanometer. It's going, as I said, through the skin, bone, and other tissue, and so on. And of course, it's more superficial brain tissue. And you have characteristic absorption bands for oxygenated, deoxygenated hemoglobin, so the blood, so oxygen-rich and oxygen-poor blood, and it's relating to the activity of these brain areas. It's very similar to the bolt signal that we had before in case of fMRI, but are confounds here. So you have also absorption due to particular processes in the cell itself that have nothing to do with your paradigm. So in slide 42, this is summarizing the different processes from light in to light out that is picked up by a sensor. So yeah, as I said, the oxygenated deoxygenated hemoglobin is what you're basically interested in, but there's also the oxidized cytochrome oxidase compound that is confounding your measurements. And then slide 43, there is the physical principle is modeled by the so-called Beer-Lambert law. There's a sort of modeling principle that you, as an experiment, are going to use to interpret the data. And on slide 44, you see the on the left-hand side, you see the spectrum in case of that is picked up by your sensor. Right? That is the part that is not absorbed. Actually, then you can you see the difference between the oxygenated and deoxygenated blood. There is some difference in the spectral response, and if you zoom into the near-infrared region between 650 and 1000 nanometer, you see some clear difference between the two cases, the two states of the blood. And of course, the oxygenated level here is of interest because that's referring to more activity of that brain area. And yeah, this is then on slide 45, the difference signal here, and that's what you can try to work on. Of course, given the presence of all these possible confounds as indicated on slide 47, you have to be aware of that. And sometimes a tracer is being used, like a contrast agent is being used to better pick up the oxygenated blood stuff and what is related to the blood flow. And but of course it's an extra complication if you need to do that. And for commercial purposes, this would not be very interesting to do, that your participants need to be injected with something like that. Okay, so it is being used in BCI. You can find some references about it. It's a non-invasive thingy, so it's in that sense interesting for commercial purposes also. And sometimes nowadays there are combinations, e.g. with NIRS. You can buy these braces or headsets that have a couple of EEG electrodes and one or two or so of these NIRS electrodes, usually placed on frontal regions where you pick up emotional responses, because that's most interesting for marketeers to pick up that. So they exist, you can buy them. And of course, it's an advantage here that it is